a variety of cancers were chosen: breast cancer, head and neck cancer, diffuse large B-cell lymphoma, 5 different childhood cancers, cervical cancer, and sarcomas. And in each of these cases, what happened was samples were collected from individuals with these cancers, both tumor and normal samples, and DNA analysis was done, either of the entire exome, all of the coding regions of the genes in these tumors and normal samples, or in some cases, the entire genome, to look at these somatic mutations that had occurred in these cancers. And by looking at a large enough number, you can begin to see which genes are affected with any reasonable frequency, and therefore must be related to the etiology of the cancer. There was an international project beginning at that time, the International Cancer Genome Consortium, of groups around the world that were beginning to do such things. I'm happy to say that this Mexican-initiated project was right at the forefront of that, and is in fact, over the last 2 years, proven to be one of the leading contributors in the world to this effort. And the Mexican-initiated project is recognized as one of a handful of the world leaders. In breast cancer, a number of discoveries have been made of genes involved in breast cancer, in particular mutations involved in breast cancer, some of which have potential therapeutic implications for patients. And this joint study between the Mexican and American scientists was published in a leading international journal, Nature. Head and neck cancer had barely been analyzed around the world. There really was very little understanding of the types of genes that were mutated in head and neck cancer. And this joint study identified many pathways and genes that were mutated in head and neck cancer, pointing to roles in certain developmental pathways of squamous cells that have really opened the door to understanding what goes on in head and neck cancer. And there are really distinctive features of this cancer. In addition, as you would imagine from something called head and neck cancer, it's a bit of a grab bag. There are many different types of cancer in the head and neck, and the ability to look into the genetic mutations is beginning to allow the sorting out of the similarities and differences between the different types of cancers in the head and neck. Diffuse large B-cell lymphoma, again, a number of important discoveries of genes related to this lymphoma, and a paper from this international consortium has been published in the Proceedings of the National Academy of Sciences of the United States quite recently. So this international team has been quite busy, but in addition, there is work going on these variety of childhood cancers. A rhabdoid tumor has been analyzed and shows remarkable features, in particular extraordinarily low mutation rates, perhaps the lowest mutation rate seen in any cancer, raising questions of how little it might take to create a cancer in some cases. And then work in cervical cancer and sarcomas is underway. So it is clear that this project has been very successful and really has established this Mexico-US collaboration as a leading force in cancer genomics. Of course, going forward, it opens the door to how can those insights into the genes that are mutated teach us about the biological pathways, and how can we use that for therapeutics. And I think as we look ahead to the years coming, it will really be the conversion of this knowledge about mutated genes into understandings of pathways and then targeted therapeutics that will be the tremendous payoff for projects like this for public health.